Gene mutation. Gene mutations are heritable change in the base sequence of individual genes or genomic DNA which may produce allelic alterations in the genotype. Since they occur within individual genes, they are also called intragenic mutations or point mutations, in distinction with chromosome mutations, that is, change in chromosome structure, and genome mutations, that is, change in chromosome number. Spontaneous gene mutations result from several processes such as DNA damage, defective DNA repair, faulty DNA replication, insertion or excision of transposable elements, etc. The frequency of spontaneous gene mutation may be increased by several factors such as 1. Defective DNA repair system, 2. Defective DNA replication system, 3. Defective DNA polymerases and DNA ligase, and 4. Production of aberrant DNA precursors. The smallest mutable fragment of the genetic material which serves as a basic unit of gene mutation is called muton. It forms a mutable or mutational site within a cistron or gene. The arrangement of mutons within a gene is strictly linear, just as the order of genes in a chromosome. Molecular basis of gene mutations. Gene mutations essentially involve abnormal alterations in the molecular organization of genomic DNA. Changes in the molecular organization of DNA are mostly due to the errors in DNA replication. This is commonly called copy error mechanism. Changes in DNA cause corresponding changes in mRNA, which in turn cause abnormal changes in the amino acid sequence of the gene products, which are enzymes and structural proteins. Abnormal changes in the gene products obviously brings about abnormal phenotypic alterations. Thus, gene mutations include frame shift, which includes insertion and deletion, and substitution mutations. Frame shift mutation or insertions and deletions. Frame shift mutations are the gene mutations which bring about a shift in the reading frame of codons through changes in the number and order of nucleotides or base pairs. They involve the insertion or deletion of one or more nucleotide pairs, collectively called indels. Indels involve one or two base can have devastating consequences to the gene because translation of the gene is frame shifted. This figure shows how by shifting the reading frame one nucleotide to the right, the same sequence of nucleotides encodes a different sequence of amino acids. The mRNA is translated in new groups of 3 nucleotides and the protein specified by these new codons will be worthless. In this figure, you will see the frame shift due to deletion mutation. Deletion of A changes the order of nucleotides in the reading frame. In this figure, you can see a frame shift mutation shifts the grouping of these bases and changes the code for amino acids. The resulting protein is usually non-functional. In general, the proteins and enzymes produced by frame shift mutations are non-functional. Hence, frame shift mutations are more deleterious than substitution mutations, other than nonsense mutations. Few examples of frame shift mutation are Tay-Sachs disease, Huntington's disease, amyotrophic lateral sclerosis, etc. Tay-Sachs disease causes a progressive deterioration of nerve cells and of mental and physical abilities that begins around 6 months of age and usually results in the death by the age of 4. The disease occurs when harmful quantities of cell membrane components known as gangliosides accumulate in the brain's nerve cells, eventually leading to the premature death of the cells. A ganglioside is a form of sphingolipid which makes Tay-Sachs disease a member of sphingolipidosis? Tay-Sachs disease occurs when hexoaminidase A loses its ability to function. People with Tay-Sachs disease are unable to remove the GalNAc residue from the GM2 ganglioside, and as a result they end up storing 100 to 1000 times more GM2 gangliosides in the brain than the normal person. Over 100 different mutations have been discovered just in infantile cases of Tay-Sachs disease alone. The most common mutation, which occurs in over 80% Tay-Sachs patients, results from a 4 base pair addition TATC in exon 11 of the hex A gene. This insertion leads to an early stop codon which causes the hex A deficiency. You can see in this figure the hex A gene is located on the long Q arm of human chromosome 15 between positions 23 and 24. Huntington disease is a progressive brain disorder that causes uncontrolled movements, emotional problems and loss of thinking ability. The HTT gene provides instructions for making a protein called huntingtin. The HTT mutation that causes Huntington disease involves a DNA segment known as a CAG trinucleotide repeat. In people with Huntington disease, the CAG segment is repeated 36 to more than 120 times. An increase in the size of the CAG segment leads to the production of an abnormally long version of the huntingtin protein. The elongated protein is cut into smaller toxic fragments that bind together and accumulate in neurons, disrupting the normal function of these cells. The dysfunction and eventual death of neurons in certain areas of the brain underlie the signs and symptoms of Huntington disease. The abnormal protein huntingtin increases the level of the p53 protein in brain cells, causing their death by apoptosis. Amyotrophic lateral sclerosis (ALS) is another neurodegenerative disorder leading to dementia and muscle weakness, occurs due to the expansion of the hexanucleotide GGGGCC in a gene on chromosome 9 from normal 2 to hundreds or even several thousand. Base pair substitution mutation. Substitution mutation are the mutations in which one kind of nitrogenous base of a codon of DNA or mRNA is substituted or replaced by another kind. Base substitution brings about corresponding alterations in the base pair sequence. This in turn brings about alterations in codons. Altered codons may code for different amino acids so that wrong amino acids may be incorporated during protein synthesis. This results in amino acid substitution in the gene product. Consequently, abnormal proteins or proteins with altered properties may be formed, resulting in abnormal phenotypic alterations. Base substitution leads to two types of molecular change: transition and transversion. Single base substitutions. Single base substitutions are also called point mutations. If one purine, that is A or G, or pyrimidine, which is C or T, is replaced by the other, the substitution is called a transition. If a purine is replaced by a pyrimidine or vice versa, the substitution is called a transversion. Missense mutation. In missense mutation, codon for one amino acid is replaced by a codon for another amino acid, resulting in new nucleotide alters the codon so as to produce an altered amino acid in the protein product, which is seen in the disease sickle cell. The replacement of A by T at the 17th nucleotide of the gene for the beta chain of hemoglobin changes the codon CAG for glutamic acid to GTG, which encodes valine. Thus, the 6th amino acid in the chain becomes valine instead of glutamic acid. For example, if a missense mutation causes a substitution of a chemically similar amino acid referred to as a synonymous substitution, then it is likely that the alteration will have a less severe effect on the protein's structure and function. Alternatively, chemically different amino acid substitutions called non-synonymous substitutions are more likely to produce severe changes in protein structure and function. In this particular figure, you can see an example for a substitution mutation. Sickle cell anemia is caused by substitution mutation, wherein the codon GAG mutates to GTG and leads to the change from glutamic acid to valine. In cystic fibrosis, the replacement of G by A at the 482nd nucleotide of the gene for the cystic fibrosis transmembrane conductance regulator or CFTR changes the codon CGC, that is arginine, to CAC, which is histidine. Thus, the 117th amino acid becomes histidine instead of arginine. Nonsense mutation. In a nonsense mutation, the new nucleotide changes a codon that specified an amino acid to one of the stop codons, which is TAA, TAG or TGA. Therefore, translation of the messenger RNA transcribed from this mutant gene will stop prematurely. The earlier in the gene that this occurs, the more truncated the protein product and the more likely that it will be unable to function. Cystic fibrosis patients bearing the premature translation termination mutations or nonsense mutation W1282X present severe pulmonary and pancreatic disease, whereas patients carrying other nonsense mutations such as G542X, R553X, S1255X, R1162X and W1316X show a severe pancreatic but mild pulmonary illness. Cystic fibrosis gene expression was found absent in respiratory tissues with mutations R553X and W1316X, which led to the hypothesis that the absence of the gene product in the lung is more favorable than the presence of an altered one. In the patients with cystic fibrosis mutations occurs in a huge gene that encodes a protein of 1480 amino acids called the cystic fibrosis transmembrane conductance regulator or CFTR. The protein is responsible for transporting chloride and bicarbonate ions through the plasma membrane. The gene encompasses over 188,000 base pairs on chromosome 7, embedded in which are 27 exons encoding the protein. Here you can see a figure on the screen. In this, the patients with cystic fibrosis, the substitution of a T for a C at nucleotide 1609 converted a glutamine codon, that is CAG, to a stop codon TAG. The protein produced by this patient had only the first 493 amino acids of the normal chain of 1480 and could not function. Silent mutation. Most amino acids are encoded by several different codons. For example, if the third base in the TCT codon for serine is changed to any one of the other three bases, serine will be still encoded. Such mutations are said to be silent because they cause no change in their product and cannot be detected without sequencing the gene or its mRNA. Splice site mutation. The removal of intron sequences as pre-mRNA is being processed to form mRNA must be done with great precision. Nucleotide signals at the splice sites guide the enzymatic machinery. If a mutation alters one of these signals, then the intron is not removed and remains as part of the final RNA molecule. The translation of its sequence alters the sequence of the protein product. Transitional and transversional substitutions. Base pair substitution mutation occurs in two ways, namely by transition and by transversion. Thus, substitution mutations are of two kinds, namely transitional and substitution and transversional substitutions. In transitional substitution, one kind of purine is replaced by another kind, that is, A is replaced by G or vice versa. But in a transversional substitution, a purine base is replaced by a pyrimidine base or vice versa. Thus, in transitional substitution, the purine pyrimidine orientation is preserved, but in transversional substitution, it is reserved. Transversional substitution is extremely rare and its genetic significance is not definitely understood. Spontaneous transitional base pair substitutions occur by three processes, namely tautomerization, ionization and binding of base analogues. In this figure, you can see the base substitutions leading to two types of molecular changes, which are transitions and transversions. 4 transitions and 8 transversions are represented in this particular figure. Tautomerization. Tautomerism is the phenomenon in which two isomeric forms of a molecule exist in equilibrium. In the case of DNA, there occurs a reversible redistribution or shift of electrons and protons or hydrogen atoms in the nitrogenous base. It is called tautomeric shift. It results in the spontaneous isomerization of the nitrogenous base to an alternative hydrogen bonding condition. Tautomeric shift alters the chemical properties of the base and causes the transformation of the normal base to a rare and abnormal isomer called tautomer. The transformation of a base from its common form to a tautomeric form is called tautomerization. Tautomerization abnormally alters the hydrogen bonding relationship of the bases. So, in the tautomeric state, a base loses its power for binding with its usual partner. Consequently, it binds with a different partner and forms an unusual base pair or forbidden base pair. For example, tautomer form of A pairs with C, tautomer of G pairs with T, tautomer of T pairs with G and tautomer of C pairs with A. Tautomerization usually occurs by the transformation of the amino group of adenine and cytosine to imino group by amino-imino tautomerization or by the transformation of the keto group of guanine and thymine to enol group, which is keto-enol tautomerization. Here you can see a figure in which lactam-lactam tautomerism is represented in the DNA. The keto tautomer is called a lactam ring, whereas the enol tautomer is a lactam ring. The lactam tautomer is the predominant form at neutral pH. The 4 bases of DNA can exist in at least 2 tautomeric forms as shown in the figure. Adenine and cytosine, which are cyclic aminidines, can exist in either amino or imino forms, and guanine, thymine and uracil, which are cyclic amides, can exist in either lactam or lactam forms. The tautomeric forms of each base exist in equilibrium, but the amino and lactam tautomers are more stable and therefore predominant under the conditions found inside most cells. The rings remain unsaturated and planar in each tautomer as seen in the figure. In DNA, adenine and cytosine usually exist in the amino form, binding respectively with the keto forms thymine and guanine through hydrogen bonding. But in their rare tautomeric imino form, they bind respectively with the amino forms of cytosine and adenine through 2 hydrogen bonds. Similarly, guanine and thymine normally exist in the keto form, binding with the amino forms of cytosine and adenine respectively. But their enol tautomeric forms remain linked with the keto forms of thymine respectively with 3 hydrogen bonds. Here in this figure, you can see the tautomeric forms of the 4 common bases in DNA. The shifts of hydrogen atoms between the number 3 and 4 positions of pyrimidines and between the number 1 and 6 positions of the purines change the pairing potential of the bases. Here in this figure, you can see the pairing between the normal keto forms of the bases. In this next figure, you can see the mispairs resulting from rare tautomeric forms of the pyrimidines. In this figure, you can see the various examples of mismatched AC and GT base pairs that can form when one of the purines or pyrimidines exist in rare tautomeric forms. The appearance of the AT pair represents a GC-AT base pair transition. The same process is also seen in the DNA double helix as shown in the figure. In this figure, you can see the mutation by tautomeric shifts in the bases of DNA. In this, a guanine undergoes a tautomeric shift to its rare enol form, which is represented by G asterisk at the time of replication. In its enol form, it pairs with thymine, and in the next stage of replication, the guanine shifts back to its more stable keto form. The thymine incorporated opposite the enol form of guanine as shown in B directs the incorporation of adenine in the subsequent replication. The net result is a GC-AT mutation. If a guanine undergoes a tautomeric shift from the common keto form to the rare enol form at the time of incorporation as nucleoside triphosphate rather than in the template strand, it will be incorporated opposite thymine in the template strand and cause an ATGC mutation. Ionization. Ionization of bases during DNA replication may also introduce transitional base pair substitution. Ionization involves the loss of hydrogen atoms from the base. Such ionized bases enter into forbidden base pairing during DNA replication. This in turn leads to base pair substitution during the next replication as exactly as in tautomerization. Binding of base analogues. Binding of base analogues is yet another mechanism of transitional base pair substitution. Base analogues are the derivatives of normal bases and they replace the normal bases in DNA, leading to base pair substitution. 5-methyl cytosine, 5-hydroxymethyl cytosine, 5-hydroxymethyl uracil, etc. are naturally occurring base analogues. Artificially synthesized base analogues include 5-bromouracil, 5-iodouracil, 2-bromocytosine, 5-methyl cytosine, etc. The first two are structural analogues of thymine, the last two are analogues of cytosine. 2-aminopurine is another base analog widely used in research, can pair with thymine, but when protonated can also mispair with cytosine as seen in the figure. Therefore, when 2-aminopurine is incorporated into DNA by pairing with thymine, it can generate ATGC transitions by mispairing with cytosine in subsequent replications. Or if 2-aminopurine is incorporated by mispairing with cytosine, then GC80 transitions will result when it pairs with thymine. Genetic studies have shown that 2-aminopurine like 5-bromouracil is highly specific for transitions. The presence of base analogues in the medium during DNA replication in all probability will lead to transitional base pair substitution. For example, if 5-bromouracil is added to the medium, it gets incorporated with a new strand in place of thymine. It may be either in the normal keto form or in the ionized enol form. The normal keto form pairs with adenine and the enol form with guanine. Enol form is unstable, so during the next replication, it reverts to the keto forms and binds with adenine. In the subsequent replication, this adenine will pair with the thymine as usual. Thus, the original GC gets substituted by AT pair. Mutation brings about genetic polymorphism in population. Genetic polymorphism is the existence of variants with respect to a gene locus, that is alleles, a chromosome structure, for example size of centromeric heterochromatin, a gene product which includes variants in enzymatic activity or binding affinity, or a phenotype. The term DNA polymorphism refers to a wide range of variations in nucleotide base composition, length of nucleotide repeats or single nucleotide variants. DNA polymorphisms are important as genetic markers to identify and distinguish alleles at a gene locus and to determine their parental origin.